Good morning, boys and girls. Here we are at the Mary Ellen Jones building. We're about to go inside and meet Dr. Kennita Johnson and hear more about the work that she does with the Dayton lab. See you inside.
Our laboratories are in this kind of new open concept. And so what's nice about it is we're right next to adjoining labs and we can collaborate, which is really nice. So the Gallipoli lab and then the Penton lab and the Dayton lab, they all do ultrasound. They do slightly different things, but because it's all ultrasound, we can collaborate and work together. And so we will go into this room here, where I have a demo all set up. So this is an ultrasound machine, and it's definitely what you think it is. It's the same kind of thing that they use for babies. And so what you get here, it starts with the transducer, and this sends out our signal and also receives our signal. And so since that's the case, it's reflective. So essentially, the signal goes out, it bounces off the surface and comes back. And so the time it takes to go and come back tells us how far something is. And then we know how far it is, we can put a pixel there, and when we put all our pixels together, that's how you kind of get the picture of the baby.
So does this give you a three-dimensional image, or are we talking about 2 dimensions here when we're talking about the pixels?
Yeah, that's a great question. So this transducer only gives us 2 dimensions, so we see what we call a slice. But there's some arrays that have rows of detectors, which is an array, and they can capture more than 1 slice at a time, or what we call a volume, or what you also know as 3D. So this is what the transducer is like. And so but our specialty is contrast ultrasound, and so we're going to do a little demo where we show you what contrast is. So this is ultrasound contrast. I'm not sure if you can see here, but it's made up of lipids. And so right now it's in liquid form. But what we're going to do is we're going to put it in this file shaker. It's going to shake it very rapidly for 45 seconds, and then we'll look at it again once it comes out. So after it shakes for 45 seconds, now we see it's a much more creamy consistency.
Looks like milk now.
Yeah. And so what that is very little tiny bubbles, which we call microbubbles. And so that's what ultrasound contrast is microbubbles. And when we go back in the room, I'll explain why.
All right, sounds good. All right, so now we're all set with all the proper personal protective equipment on.
Yep.
Now we're ready to do some science.
Yep. So we have to get the bubbles out of the vial. So we always give them a little shake by hand before we start. We put in 1 needle to vent. You hear the little squirt there. And then we use 1 needle to collect into the syringe. And so part of the reason we do that is because they are indeed bubbles. And so essentially what happens is if you're taking bubbles out without putting air in, the bubbles collapse. So I don't know if they it's kind of like if you have a can of juice, you know, you have to put a hole on each side for it to be able to get out, or a can of oil maybe. And so here is our solution here. And so what we have set up is a transducer like this that's in water. So why is water important? Where our bodies are made of mostly water. And so like I was explaining before, this not only emits the signal, but it gets the reflection. Mm-hmm. And so water and air are very, very different. And so it can't go through air very well to get to our body, to the water. So what you'll notice, what you'll see sometimes, or most of the time, is that we put ultrasound gel. So you put the ultrasound gel down, and then you put the transducer down, and that acts as, eliminates the air in between, and it lets, this is a lot like thick water, mm-hmm. and so it matches the water in our body. Mm-hmm. And so the ultrasound signal is able to go through this into the body, take the picture, and come back.
Awesome.
So, but we say that air is not something that we want. Well, in the microbubble sense, it is. We can use it to our advantage. And so these are tiny little bubbles that we normally inject into the vasculature, and we can see where blood flows. And so this demo is just going to tell us a little bit about like what it looks like, mm-hmm. and then we'll go through some examples when we see it in our different models.
I see. So now I recall hearing at some point in my life that it's dangerous to have, you know, gases injected into your veins and your arteries. Is this safe, and how is it different from that situation?
It is safe.
Okay.
The bubbles are very, very small. They are about the size of a red blood cell, maybe even smaller. And so the total volume of air is not that much, and therefore it won't harm you.
Cool.
So it's safe. It's used in the hospital, especially in heart imaging. It's really mm-hmm. cool because you can see it kind of swirl around. Mm-hmm. But it is safe.
Okay.
So what we have here is that same transducer. I've placed it in water. And so that water is kind of like our body. Mm-hmm. And a lot of times when we do our experiments with just looking at the bubbles and the dynamics of bubbles, we will do experiments just in water. And so if we look here, this is what the image is coming from that. You may be able to see, let me turn the lights down a little bit.
Yeah, I was going to say, maybe if you turn the lights down, it would help. You can kind of see things. But, oh yeah, there we go. That looks great.
Okay.
These lights are on a dimmer?
Uh-huh. So you can kind of see that there's these little things falling. Mm-hmm. And those are some air bubbles that are just in the water. And so a lot of times when we're doing our experiments, just looking at the microbubbles themselves, we don't want these other bubbles around, so we do what we call degas the water, mm-hmm. and we take those bubbles out. And so because of this, we know that this is not degassed. Mm-hmm. But this is just the water. So what we're going to do now is to inject the contrast agent in and watch kind of what happens. So I think you can see mm-hmm. this light area here. And so the contrast agent allows us to, there's some more, allows us to see more and control that. Mm-hmm. And so essentially when we say contrast agent, it stands out compared to the background. Mm-hmm.
So now I can see how if you know, we're injecting this into some organ or some tissue, and there was a path for these microbubbles to flow, then it would highlight that path.
It will highlight that path,
okay.
Exactly. And so
Yeah,
that's what we get there. Yeah. And it is, you're right, there's a lot of bubbles. And so now they're just kind of swirling about in the water. And we'll talk about some of the measurements that we take, but one of them is how long do they circulate in the body? So the body normally takes them out by removing the gas through the lungs. And then that lipid shell, remember, it started off clear. That's still there. That's what's making the bubble itself. That gets cleared out by the liver. And so they only persist or stay in the body for about 10 minutes.
Okay.
So it's not something that happens, it's not something that takes a long time. They clear pretty quickly.
Cool. So for the budding biochemists out there, can you tell us what kind of lipids you all use, or is that proprietary? I don't know if it's like a trade secret or not.
That part is not proprietary. That part, it's not a secret.
Okay.
But what I will tell you is that one of the things that is nice about these bubbles is, especially for the budding biochemist, is that we can add things to the outside of the bubble. And so one of the things that we can do is look at the molecular environment. And so if we add some sort of ligand that will target a specific site, instead of them just kind of circulating and moving like this, they'll stick in one place. And then they stay they stay longer than the 10 minutes.
I see.
And so if that receptor is there, then we know from these, what we call targeted bubbles, that there's some sort of activity like angiogenesis, or we'll look for like specific factors like tumor necrosis factor or something like that.
Okay. So angiogenesis, could you remind us what that is?
So angiogenesis happens I guess it happens in other conditions other than cancer, but it's the growing of new blood vessels. And so it's very important in cancer because as cancer grows and it needs more food, they grow vessels really, really quickly. And they're very, what we call leaky. So we try to take advantage of that as well. And so we look for markers of angiogenesis. Is this tumor asking for more materials, signals saying I need to make more blood vessels? We have some bubbles that start off in liquid form, which are much smaller, and then they can get out of the blood vessel, and then we can turn them back into a bubble with ultrasound. And then we know that vessel is like what we call leaky. I
see. Okay, so wait a second. That sounds like magic for a second. So you're saying you'll have this microbubble suspension, and they start off smaller, and you can use the ultrasound to make them expand. So you're heating up the gas inside the bubbles to make them expand?
We're increasing the pressure.
Increasing the pressure, okay. Okay, so you increase the pressure, you increase the volume.
Yes.
Okay, and the temperature should stay about the same, perhaps.
Yeah.
Okay, got it. Okay.
And so essentially what we're watching now is just, you can kind of see here where they're all kind of falling out, but all of them are just kind of swirling around. And so I will show you pictures of kind of how we use this as well. So now we're going to look at kind of microbubbles in action, how we would use them. So my research is on kidney disease, and the idea is how can we figure out which kidneys are going to get kidney disease early in the process so that perhaps they can be treated and it will prevent patients from going on to get having to do dialysis or kidney transplant to stay alive. And so the earlier we can figure that out, the earlier we can kind of start treating. So how do we use microbubbles for this? So there's a couple different ways, but this is one way in particular. So what we have here is a small animal model, and it's the same animal under 3 different conditions. And so what happens is we inject the contrast, it breaks, and then we watch the contrast flow back in. So why is that important? We're looking at how fast are they flowing back in? Is there any spaces where they don't flow in? And so this is called perfusion imaging, to see if the whole tissue is being perfused and being perfused equally. So basically what we have here is the bubbles break. You see that gray, and then we see the bubbles flow back in. So the outside of the kidney is the cortex, the inside is the medulla. And we yeah, this is better. Yes. Okay. And so this kidney on this side is the rat under normal conditions. And so as this cycles back around, you'll see that gray, that's when they break, and then you see them pour back in. We gave the animal angiotensin 2, which constricted the vessels, and then we watched how the animal reacted. So in the second one, we see it's a little bit slower than the first, and then the last that got the most angiotensin it's the slowest of the 3. And so we expect them to flow back in at a certain rate. So we always have what we call control animals, and that's the normal condition. And then we compare that to some sort of impaired version. So either a high dose group, or in our case, a diabetic group, or in this particular experiment, it was the animal served as its own control because we could add the angiotensin and watch how the kidney reacted. So we can compare this first to this last.
I see. And so what sort of disease states or pathologies are associated with elevated angiotensin?
That is a good question. So we know that high blood pressure is, and we know that along with diabetes, high blood pressure also causes kidney disease. And so it's important to understand what excess angiotensin may do to the kidney and what it looks like under ultrasound. And one of the things that we also do, which I probably should have mentioned, is we take these and convert them into math. So we essentially figure out, we take a region of interest, and then we measure how the pixel values change over time, and then we fit that to an equation. And so when we compare the coefficients of the equations, then we can kind of get a measure of impairment.
I see. Very cool.
And so this is the small animal model. So a lot of times we start with small animal models because we have a lot more control. So for instance, we can give them angiotensin. We can produce diabetes in an animal, and we know exactly when diabetes is starting because we induced it. So again, it allows us to have an animal serve as its own control because we can do kind of preconditions versus post-conditions, which is nice. And it's very controlled. We also do large animal models. So we've imaged cats and monkeys. And then I have a clinical partner that does human imaging. And so this is what a human image looks like with using the same kind of imaging method. So as we see, the human kidney looks very, very different. And this should loop around. And so we'll see the break. We see that there's certain areas that are not filling in. So that may be an indication of a lesion or like dead tissue. That blood's not going to that region. We watch it again. That gray is the breaking of the bubbles. So the ultrasound can break the bubbles, and then we watch them flow back in. And then that's how we take our measurements. But we also kind of look at, well, where are they flowing back in? Are there any problems?
And you said, did you say if this patient had a particular disease or not that you know of, or just?
A lot of times we're blinded, but we'll report to the clinical partner and say, all right, these specific sites are of interest to us. Can you give us more information?
Okay, that makes sense. That sounds like good science.
Yeah.
You don't want to bias it in any way.
Yeah.
Right? Okay.
And so part of it is just the complexity of the kidney. So where a rat kidney is kind of this one singular kind of intermedullary area, the human kidney has these individual pyramids. And so my job as a biomedical engineer is, how do I take this simple version that I use in my lab, and the point of using this simple version is to figure out how are we going to image this, what information can we get out of these images, and then to translate it eventually to people and be able to get the same information with a more complex structure.
I see.
And so that's my job as a biomedical engineer, is to figure that out.
Yes. And so that's where your coefficients and your equations come into play, right? And trying to like sort of scale up and transform them to match what's going on with the human kidney.
Yeah.
Okay.
And we also understand too, this is just one part of kind of the overall tools that the clinician uses to diagnose. So this goes along with blood and urine, physical exam. We also we're looking into other imaging to see if it can also provide some other information. The one thing that's nice about the ultrasound is that because the microbubbles are about the size of a red blood cell, they stay in the vasculature in that particular form. And then what happens is they don't get out of the vascular space in the kidney, which causes some contrast-induced nephropathy, causes the kidney to kind of shut down. And so it's still controversial, but essentially what ends up happening is a patient goes in for something else and they get a CT scan or an MRI and they give them contrast agents for those particular imaging modalities, and then their kidneys shut down. And then they're like, oh, there was a problem with the kidneys.
Oh, no.
And so what's nice about this is that microbubbles don't do that. They don't stay around that long. They go in the kidney and they get out of there. And so you don't have those same kind of issues.
Wow, that's scary.
Yeah. Yeah.
Hello, Dr. Johnson.
Hello.
Thank you, first of all, for spending the time with us this morning, and being able to take us on a lab tour and show us, all the really incredible work that you're doing. Can you tell us a little bit about your background and how you ended up here at UNC?
Yes. So I grew up in Prince George's County, Maryland. I went to Eleanor Roosevelt High School, and then I went to UMBC, University of Maryland, Baltimore County, for undergraduate, where I majored in physics. And there I was a Meyerhoff Scholar. So from there, I think when I went into college, I just knew I was going to be an astronomer. Like, that's all I wanted to ever be. And then I think I took modern physics, and I was like, what do they do? I don't want to do that. So most of my friends, most of the Meyerhoffs were going off to be MDs. And so I had no desire to work with people or see blood or anything like that. And so I found out about medical physics. And so that's what took me to the University of Florida. And I got into their medical physics program. And then once again, I found myself going, is this what we do as medical physics? And so my advisor said, look, maybe biomedical engineering is maybe more your speed. You can do probably a lot more physics than you actually do as a medical physicist. A lot more computer programming and all the things that you really want to, the things that you're good at. And I think the thing that really ties together both the astronomy and the kind of the medical physics was the images. So I really like working with images. And I really liked working with medical images, actually. Mm-hmm. But it's frowned upon to work on people's medical images in the hospital. And so I was lucky to get a postdoc learning more about small animal imaging. And that postdoc was here. I'm in the Triangle at the National Institute of Environmental Health Sciences, NIEHS. And so there I was in a pathology lab. It was a great time. So one of the things that I learned to do there is understand a lot more about biology. I had to do my imaging without kind of getting in the way of other experiments, which was nice. So learning a lot about non-destructive imaging. And so even after the postdoc, I knew I liked images. I got to do a lot of image analysis work. So all that computer stuff that I really liked doing. I found out I really liked conducting studies. I spent a year like consulting, just like doing image analysis for other people. If they had, and when I say medical images, I'm talking about CT images, MRI, ultrasound, nuclear medicine. Okay. So all the different kind of images.
So not like the gross anatomy stuff where you're looking at.
No.
Okay,
no. Okay. More kind of internal images. And the idea being understanding which image provides information about what organs. Mm-hmm. So kind of really understanding those aspects. One of the things I learned as a postdoc, it's like, how do we take the image that we need? So like the parameters, the angles, like all of that, so that we can get to the answers that we eventually want. So spending a year consulting, just doing the image analysis, the end part, wasn't really satisfying.
I see.
And so I took a job here on campus as a research associate. Mm-hmm. And so a research associate is just a person, you have a PhD, but you're just doing research. You're not doing, you're not necessarily conducting your own research.
Mm-hmm. You're not teaching.
You're not teaching.
Right.
No, so you're just kind of every day doing research. And so I did that in the Dayton lab. I started here, and that lasted for, I guess, about 5 or 6 years. But in the same time period, I moved up to lab manager. Our lab bloomed. We had a bunch of people, and so then it's the concept of how do you manage a group of high functioning researchers and making sure all research is getting done and people aren't crossing each other, and so that was really good. It was really good for me to understand that kind of management aspect. Mm-hmm. And so from there, it looked like I started developing my own ideas. I picked up a nephrology partner, Emily Chang. She's a nephrologist at the Kennita Center here. And we applied for our first grant. And so $2,000 in a dream. We got that $2,000 and we started taking kidney images. And then we started getting more and more grants. And so I moved up to a research assistant professor. Mm-hmm. So a research assistant professor, you're responsible for research. You're not responsible for teaching. The caveat being that because you're just responsible for research, you're responsible for funding yourself. So that means that you have to get enough grants and do enough research to pay for yourself. And that actually worked out very well. So we started, we really started to collect the grants and start doing research. At the same time, I did start teaching actually at North Carolina A&T. They have a biomedical engineering program there. they needed someone to start teaching their imaging course. So that's where I started. And so for the last 6-7 years, I've been teaching there every spring. So
very nice.
I love it there. I love the students there. And I love kind of getting to know them and encouraging them to go to grad school. So more than likely, if I see you, I'll say, have I talked to you about grad school? Just because I think it's such, it's so important. And so then it bounces back to here because of the work that I've been doing there at North Carolina A&T. They've made me a director of diversity and equity in the biomedical engineering department.
Awesome.
And so, so I spend part of my time working with A&T, but also just trying to get more students of color, LGBTQ, students with disabilities into our department so that we can, because we really need people that come from all of these different points of view to help us kind of tackle all these different diseases out there. And biomedical engineering really comes from kind of your point of view, you bringing your creativity, your experience to any project, because a lot of times we do work in teams and that really spurs innovation. Yeah. So I would highly recommend biomedical engineering to anybody who is interested.
Yes, we'll definitely have some students applying. Yeah, can you tell us just really quickly a little bit more about your specific research interests and what sort of problems you're excited to tackle these days? So you say you started getting into kidney research.
Yeah, so kidney research always interests me. I had a grandmother that had diabetic kidney disease. And so the idea, so my grandmother, she was very, she adhered to everything they said. She took all her medicine, she did all the exercises and all that, and still didn't prevent her from getting to dialysis. And so the idea is, how can I take the talents that I have and apply it to problems that affect my community? And so one of the things that we're interested in is we think that ultrasound will tell us something about the kidney environment. Because essentially what happens is the environment changes, and then damage happens, and then it shows up in blood and urine. So if we can get it at the environment changing stage, we can use medicines to maybe put the environment back to where it should be and hopefully prevent damage that goes on to lead to dialysis.
Okay. And
so, and part of the reason we picked ultrasound is because it is safe. The contrast agents are safe. They stay in the vessels. They don't have the same problems as other imaging contrasts. It's really portable, so you can have ultrasound machines kind of anywhere. They wheel around from place to place. They're even getting to the point now where people can just plug a transducer into like their iPhone and take pictures. Wow. So, they're really portable, so and much less expensive. So if we think about taking them to clinics or communities, or, like when you have one of those things where you set up and you take people's blood pressure and check them for diabetes, our dream is that let's just see how your kidneys are functioning as well. Yeah. And so, so just kind of thinking about the problem as a whole, taking the expertise that we have and applying it to something that would help my community is really kind of what drives what I want to do. And so, since I have grown, in the fall I'll be getting my own lab.
Congratulations!
Yeah.
That's awesome.
So I'll be moving out of the Dayton lab into my own space and conducting my own research. And so we'll still be continuing with our kidney projects because those are kind of taking off. But we'll also be looking at maternal health because that also affects Black and brown people, women, more than their Caucasian counterparts. So as we know, ultrasound is safe for, babies in utero. So that it should be a tool that we can use to really help us to understand what's happening with the mother.
Very cool. Well, Dr. Johnson, thank you so much for your time. That's a really inspiring story that you shared with us, and so I really, really appreciate that. And I'm very excited to work with you and to send our students your way as much as we possibly can.
Definitely.
Thank you so much.
Thank you.
All right.